The following slide, slide 5, is a description of the 4 primary subtypes of epithelial ovarian cancer. And these subtypes are classified by histopathology. What these tumors look like when they're removed from women and examined by a pathologist. And so these, what you see in this diagram here, are sections of tumors that have been removed from women. And the pathologist then can classify those types of tumors based on what they look like. And you have 4 lined up in front of you. The first one is classified as serous, the second one as endometrioid, the third as mucinous, and the fourth on the far right is a clear cell carcinoma. And I have no idea what kind of audience I'm dealing with or your areas of expertise, but I think even I, who has no experience in pathology other than our own research, can see that there are some very distinct differences in the appearance of these tumors. And they not only look different, but they behave differently too. And so the response of patients to the standard care, which is surgery followed by chemotherapy, often a combination of carboplatin and Taxol, does not always have the same result in women with these different types of cancers. And so they look different and they behave differently. And of course, what the research wants to do is to find out what caused them to become so different in the first place and how we can take advantage of those differences to be able to treat them differently. And so we are having a rapidly increasing derivation of information from these different types of cancers to tell us that, for example, serous cancers, the first one shown there, very commonly has mutations in a tumor suppressor gene called P53. And that makes those tumors really quite aggressive. Endometrioid cancers seem to very often have a mutation in a tumor suppressor called P10. And so on. And so these each have characteristics that are quite distinct, not only by looking at the cellular arrangements, as you see in this slide, but also at the molecular level, the mutations in tumor suppressors or the activation of proliferating pathways that are contributing to the way they behave and the way they respond to chemotherapy. And so understanding the molecular differences between these is a very active area of research around the world. And to complete the introduction is slide 6, and this is just to highlight some of the mechanisms, some of the methods that we use to study ovarian cancer, and they are shown progressively from left to right, where we have procedures that use what are called in vitro methodologies. These are cells or cell lines that have been taken from an ovarian cancer and placed into culture, and we can observe and test these cells when they are in culture. You can see the second slide from the left actually shows a very nice epithelial ovarian cancer cell line. These are cancer cells that have been dissociated from the original tumor, placed in culture, and allowed to grow and can be used extensively for studies. We are using in my lab right now cell lines, a large panel of cell lines, some of which were developed back in, we checked yesterday, it was 1982 when some of these original cell lines were developed. And we are still doing very active research with those same cells as they continue to grow in our lab, and we can use some of them for research on a continuing basis. And we've learned a lot from cell lines, but they have their limitations. And one is that a cell line, when it's plated in a culture dish and is growing in 2 dimensions, actually doesn't behave completely the same as one would in a tumor. And there are components of tumors that simply are not present when you have these cell lines in culture. And so one step towards being more similar to what's found in vivo are 3D cultures or the formation of spheroids. And Dr. Mesmasson's lab in Montreal actually has been one of the pioneers in growing the ovarian cancer cells as spheroids or aggregations of cancer cells. And it's that 3-dimensional structure that begins to have them behave quite a bit more like human ovarian cancers. And part of that is the associations with cells are similar to cancers. These, however, and if you look at the gene expression in these spheroids compared to the primary cancers, they are much more like the human cancers than they are the cell lines in culture. And so we do know that they are behaving a little bit more like the human cancers. There are cell components of a tumor, however, that are not present in spheroids or in cell lines. While these epithelial cancer cells represent the epithelial component, most cancers have at least two other components, one being the stroma and the other being blood vessels. And so if you truly want to study a disease that replicates ovarian cancer better, then we move from spheroids to actually studying either the original tumor tissues, so samples that have been taken from women who have had surgery to remove their ovarian tumors, and those samples come to us for research purposes. And then the final category, and one that we have worked on extensively in my lab, and that is the animal models, either what's called xenografts or transgenics. And these models are actually quite valuable from my perspective. Xenografts are created by taking human ovarian cancer cells and injecting them into mice that are immune deficient, so that these tumors, these human cancers, can grow in these mice, use blood vessel development and interaction with stroma in a manner that actually resembles human cancers quite well. And they have been used extensively for therapeutic testing, so almost all companies, biotechnology and pharmaceutical companies that are developing new drugs will use xenografts as a method to test whether the drug will work in an animal model. So in an animal model, in this case, that is expressing or is developing a human ovarian cancer. And the final category is transgenic animals, and this is where my lab has really been part of the forefront in the world for developing these models. These are animals in which the normal ovary has been manipulated to develop into ovarian cancer, trying to keep as much as possible the same changes that occur in human cancers. So mice do not develop ovarian cancer in the same way that humans do, but with some small genetic changes, we can make those mice develop tumors that look and behave very similar to human ovarian cancers. And so that allows us to study the process of how normal ovary becomes cancer. And so these are the models that are used currently for fundamental or basic research, and I just thought you might be interested in seeing how research is done with these various systems. So moving then on to slide number 7, we begin to talk about the types of research, and it's essentially divided into 4 different categories, 4 different types of research that takes place. And you can see how if you were going to do all of these in great detail, we'd be here for hours and hours and hours, and that would be okay with me, but I'm sure some of you have better things to do this afternoon. So we're going to talk about the highlights of each of these categories of research. And the first one is epidemiological. This is the research that investigates risk factors, factors that may be involved in the prevention of cancer, largely dealing with population bases or dealing with people. So studying people, either people with ovarian cancer or people who are at risk of ovarian cancer, to see if there are factors in their lives or that they are exposed to that might help to either identify who is at risk or even better find ways that we can prevent or reduce the risk of the cancer entirely. And so I'll give some examples in a few moments. The other type of research is called clinical. These are largely the physicians who are involved in doing clinical trials, for example, where they are testing new cancer treatments, new therapeutics, new devices, doing tests of any sort that would help, that are working directly with the patients, and usually the patients with cancer, to see if it can improve the outcome for their disease. And so these are clinical types of research. Translational research is actually taking work that has been done in the lab and trying to translate it or to transition the discoveries into the clinical environment. And so, for example, I mentioned that xenograft models of mice are often used to test new therapies. So a biotech company that might come to us and say, we have this new drug that we think might work on ovarian cancer. Can you please test it and see if it has any impact on your animal model of ovarian cancer? We will do that, and if it turns out that that new drug or that new approach has any benefit at all to the mice, then the company has some background, some evidence that it can take forward to further developing that drug towards phase 1 clinical trials, the first step of testing that new drug in patients or in people. And so that type of research actually sounds easy, but it is the most challenging. Taking something from a laboratory discovery into a clinical environment obviously requires a lot of major steps. Requires in Canada, Health Canada involvement. Research ethics boards need to be involved to ensure that the drug will cause no harm or that it will minimize the risk of causing harm in the initial stages of investigation. So it is probably the most important step and the one that tends to be the slowest and the most challenging. It really does involve a large team of individuals with different areas of expertise. And finally, the fundamental or the basic research is the research that most scientists would do. When you have PhD scientists working in research, they tend to work on the fundamental basic, or although there are many like myself who work very closely with our clinical colleagues to try to facilitate translational research at the same time. And so the fundamental basic research is basically looking at what is causing this cancer, and the cellular events and the molecular events. What is going on inside the cells that is making that normal cell become a cancer cell? So if we go to the next slide, the emerging themes, and there are a number of them, which is a very promising sign, and these are all terms or phrases that have come up within the last few years. If you had said cancer stem cells about a decade ago, nobody would know what you were talking about. Cancer stem cells are thought to be a cellular component of most cancers, and that cellular component is thought to be very resistant to chemotherapy and radiation therapy. It has really excellent survival mechanisms, and so those cells tend to resist death, while all the other cancer cells around them might be undergoing death because of exposure to chemotherapy or radiation therapy. These stem cells seem to be pretty resistant, and they are thought to be the source cells when the disease recurs. And so there's a lot of research in trying to identify the characteristics of these stem cells that make them so resilient, but also then what can we do to make them less resilient, to make them more responsive to either chemotherapy or radiation therapy or some other treatment that might be able to eradicate them. Circulating tumor cells has been in the news again very recently because it has been found by a group at Georgia Tech that they can take a very small volume of blood and detect a single cell among a single cancer cell among the billions of normal cells that are present. This is very high sensitivity, obviously, to detect a single cancer cell among this blood sample and all these normal blood cells that are present. And so the question is, what can be done and what is being done to use the ability to measure these circulating tumor cells? And one of the clear benefits that could be implemented right away, if this technology could be adopted by other institutions, is to be able to use this to monitor cancer progression. We do have, for most cancers, a way of showing that a specific treatment has had a response, but this one is far more sensitive, I think, than anything that is out there. And so if you could measure, take a blood sample just before chemotherapy is administered, for example, and then take blood sample measurements periodically after the treatments have begun, should be able to see that there's a decrease in the number of cancer cells that are detected in the blood, and then use this to monitor when the tumor might recur, because there should be then an increase in the number of cancer cells that are found in these blood samples. This is not a simple technique yet. It's just been sensitized to the point where it can pick up a single cell in a blood sample, but it is not an easy analysis yet to do. But it is a very exciting opportunity, and ultimately down the road it would be interesting to see at what point in the first stages of a primary cancer do these tumor cells appear? Do they appear at early stages of ovarian cancer, for example, where we might be able to use them for early detection? So this is all being investigated at the moment.